Hello. So very good evening to the students who have joined. Can you please let me know in the chat box if my voice is audible and my video is visible to you? Also if the presentation is clear. Let me know in the chat box, please. Alright, so we will wait for some time and then we will start today's session. So we will wait for a few more minutes, then we will start today's session. Meanwhile, if you have any problem while seeing the PPT or hearing my voice, please let me know in the chat box. Okay. Okay, so I think we can start with the introductions. Okay, so very good evening to all the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT Dhanbad. Today it is the week 3 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. So today it is the week 3 live session where we will discuss about the course content which was in your week 4 for the same course. Okay, in the first week we did in the first week's live session we completed week 1 and week 2. In the second live session we completed the week 3 course contents. So today in the week 3 live session we will take care of the week 4 course contents. Okay. So I think my voice is clear. So if you have any problem in hearing my voice, please let me know if it is not clear or anything. And also I hope my presentation is all visible to you. So like every day we will today discuss about 20 questions related your week 4 course content. Okay, when I show you some question, please try to answer the question in the chat box. Your answer may not be correct or it may be correct also, but whatever it is, please do try, because we don't have any marks regarding this, but if you at least try, it will be more interactive session. And if you have any doubt, any query, anything, you can write in the chat box. Okay, so I think we can start the session. So first of all, I want to wish all of you a very happy Independence Day. So it is a holiday, but still we have to do our session, so bear with me for next 2 hours. So before starting, I want to brief a few points like every day. So there will be a problem solving sessions every week, and this session will be on Tuesday, every Tuesday from 6 p.m. to 8 p.m. and the sessions will be live on the Google Meet platform. If any network disruption happens, please wait for a few minutes. Do not leave the session. If I face any kind of network issue, I may rejoin as soon as possible, but you don't leave the class. And if the during the class you at your side the network problem happens, so don't worry, because we will share the today's class lecture and also the PPT with you soon. Okay, and the chat box is always open. If you have anything to tell me, you can write in the chat box. And if after class you have any doubt or any query, you can drop an email to me at the end of the session. I will share my email ID to you. Okay, so at the end of the session I will share my email ID to you. You can just write an email to me, and the presentation and class recording, as I have said, it will be shared to you soon. Okay, so if I hope my voice is clear, right? And you can see the PPT also. Please confirm in the chat box. So the chat box is always open, right? So I want all of you to respond me through the chat box, and also when I show you some problem, Please, yeah, very thank you. So if you have any problem, any doubt, please let me know in the chat box. Also, please try to answer the questions which I show to you. I will discuss it in detail later. So don't worry if your answer is wrong or if it is right is very good. So today in this week 3 live session, what we will do, we will discuss our week 4 course content. So what is in the week 4 course content? So it is the enzyme kinetics, which will be continued in this week, because in last week we did start about the enzyme kinetics. We did a few questions related to the Michaelis-Menten enzyme kinetics, and also what is the induced fit hypothesis about the enzymes, what is the enzyme. We did a brief about it. So today we will continue that enzyme kinetics, and I think we will solve few mathematical problems today. Okay, and then we will move forward to the concept of the enzyme inhibition, and it will be continued problems on enzyme kinetics and enzyme inhibition. As I said to you, I will show you few mathematical questions today, and I hope if you can solve the question today, or if you at least try to solve, it will be helpful to you for your assignment purpose. Okay, then we will move toward the synthetic biology, and later in today's class. So I think we can start class. Meanwhile, if more students join, I will just let them in. Okay, so let us start. So the first question in today's section is, what is the purpose of having 2 aspartate residues in the aspartyl protease? Your options are: number A, 1 aspartate helps the release of hydrogen ion from water, make hydroxide ion. The other aspartate in turn gives stability to the oxyanion that is being generated. Option B, 2 aspartate residues make the active site more flexible. Option C, none of the options A or B are correct. And option D, both of the A are B and correct. So try to answer this question in the chat box. What is the purpose of having 2 aspartate residues in the aspartyl protease? Try to answer it in the chat box. Which of the following 4 options you think is correct regarding this question? After your trial, I will just show you the answer, and we will discuss what is aspartyl protease and what is its role and how the active site works. What is the purpose of having 2 aspartate residues in the aspartyl protease? Number A, 1 aspartate helps the release of the hydrogen ion from water, make hydroxide ion. The other aspartate in turn gives stability to the oxyanion that is being generated. Option B, 2 aspartate residues make the active site more flexible. Option C, none of the options A and B, and option D, both A and B are correct. So is there any answer? Okay, so I want all of you to try. At least your answer may not be correct, it's fine, because we don't have any marks on this, but I think you should try to answer this question. I will give you few more minutes, then I will show you the answer and we will discuss. Okay, so I think we are getting few answers, which is number A. Okay, let me show you what is the answer and then we will discuss it. So what is the purpose of having 2 aspartate residues in the aspartyl protease? The correct answer is, very good, it is number A. 1 aspartate helps the release of hydrogen ion from water, make hydroxide ion, and other aspartate in turn gives stability to the oxyanion that is being generated. So what is the What are the proteases? So proteases are the member of same family, are usually eventually related. So proteases is a type of enzymes. So there are several kind of protease based on their active site mechanism. Okay. So there are serine proteases, where serine, amino acid serine, it actually plays a very important role. What is the examples of the serine proteases? These are trypsin, chymotrypsin, elastase, thrombin, subtilisin, factor D, factor B, etc. Aspartic protease or aspartyl proteases are the protease where the aspartate ions or the aspartic acid residue in the active site, they play a very crucial role. What are the example? These are gastrixin, capsaicin B, renin, pepsin, HIV protease, etc. Metalloproteases are those, the examples are the thermolysin, carboxypeptidase, matrix metalloprotease. So these metalloproteases, they have a metal in their active site, okay, and this metal ion, they play a very vital role in the protease mechanism. And there are cysteine proteases like papain, calpain. These are the cysteine proteases, where the or thiol protease we can also tell them, where the cysteine moiety is very crucial. Okay. Okay, so since I have made the voice and video off for the students, so that there is a smooth streaming. So if you have anything to tell me, you can write in the chat box. Okay, I can see there is a raised hand. So Pranita, if you have anything to tell me, you can write in the chat box. Okay. So let us move forward. So I was discussing about this aspartyl. So as you can see, this is the active site for the aspartyl protease. Okay, this is the enzyme active site for the aspartyl protease. As you can see, there are two aspartate residue. One is here and another aspartate residue is here. Okay, this is aspartic acid residue, means this residue is in the acid formation, and here as you can see, it is a carboxylic acid formation. And as you can check, let me just erase it a bit. Yeah. And in other case, the aspartyl residue, it is a aspartate or carboxylic acid, carboxylate ion. Okay. So in first step, what happens, this aspartate residue, it takes up the hydrogen atom from the water and make it hydroxide, which is a very more strong nucleophile than the water itself. Okay. This hydroxide nucleophile, that attacks this peptide bonds, the carbonyl carbon of the peptide bond, and there is a oxyanion which is formed here. And what happens, there is another aspartate residue here, and this aspartate residue has a carboxylic acid group. This carboxylic acid group, it gives the stability to this oxyanion by the in the mode of hydrogen bonding. Okay, so here there is a hydrogen bonding present, and through this hydrogen bonding, this particular oxyanion, it gets stabilized. Okay, and after that, what happens, there is a hydrolysis happens and we get this enzyme back. So what is the role of the two aspartate ion in this case? Then the first aspartate ion, what it does, it actually helps the release of the hydrogen ion from the water, making it hydroxide ion, which is the more nucleophilic and a better nucleophile. That hydroxide ion attacks the carbonyl carbon of this peptide moiety, and the peptide bond hydrolysis happens. So by this method, the particular peptide bond hydrolysis takes place in this particular case. Okay. So it was the example for a aspartyl protease. Okay. Now if you have any doubt, anything to ask, you can write in the chat box. Otherwise, I will move toward the next problem. Okay. Okay. So now I will move forward to the problem number 2. So the problem number 2, it says that which of the following statement is true regarding enzymes and receptors? Option A, only receptors have active site, not enzymes. Option B, only enzymes have active site, not receptors. Option C, after binding, reaction takes place in both enzymes and receptors. And option D, after binding, no reaction takes place in receptors. So which of the following statement is true regarding the enzymes and receptors? So whenever there is a true false kind or incorrect correct incorrect kind of question, please read the question and options very carefully, because this type of questions sometimes they may be a little bit tricky. So but if you read the options carefully, the question carefully, it will be very easy for you. Okay, so here read the options carefully and identify which of the following statement is true. Your option A is only receptors have active site, not enzymes. Option B, only enzymes have active site, not receptors. Option C, after binding reaction takes place in both enzymes and receptors. And option D, after binding no reaction takes place in receptors. Try to write the whatever you think is the correct answer in the chat box. As I say, there is no marks regarding this, but I think all of you should try to answer this kind of questions. Okay, it will only help you to understand the course better and also in your weekly assignments. Okay, let me check if there are any answers. So I have few answers for it. We have number D, number C. So I have 1 number D and 3 number C. Okay, let me check which is the correct answer. So the correct answer is number D. After binding, no reaction takes place in receptors. But we all know after binding of the substrate in the enzyme, there is a chemical reaction or any kind of reaction and products are forming. But in case of receptors, no such reactions happen. So what is the difference between enzymes and receptors? In receptor, there will be an area which has a correct shape to accept the incoming messenger. This area is known as the binding site and is analogous to the active site of the enzyme. Chemical messenger do not undergo chemical reaction. It fits into the binding site of the receptor protein, passes on its message and then leaves unchanged. So chemical messenger is something like a substrate to an enzyme, and chemical messenger is the same for the receptors. Okay, they actually bind, these chemical messengers, they actually bind to the receptors, but there is no chemical reaction happens. Okay, and the chemical messenger, they leave the reaction site or active site of receptor unchanged, unlike the case of substrate and enzyme. When the substrate binds to an enzyme, it reacts and the products are formed. Okay, so that is a major difference between the receptors and the enzymes. So some receptors characteristics are: binding of the ligand is only temporary. Ligand binding produces physical changes in the protein conformation, initiating intracellular changes that ultimately generates behavioral effects. Receptors have a life cycle as other proteins do. Receptors can be modified in numbers, long-term regulation in sensitivity. Receptors can upregulate increases in number, the chronic absence of agonist, and downregulate decrease in the number, chronic presence of agonist. So these are few characteristics for the receptor molecules. We will discuss about these receptors and what are the exact types of receptors and what are the roles later in today's class. Okay. But it is very good that all of you, at least many students, try to answer this question. So it is very important, as I have said. So if you have any problem, any question, you can ask me right now, or you can just ask me later at the end of the session. I will move toward the next problem now. So problem number 3, it says that which of the following statement is true regarding the thiol proteases. I have mentioned about this thiol proteases in the first slide itself, like in thiol proteases. So the options are: number A, it can react only via catalytic triad of aspartate, histidine and cysteine. Number B, histidine is not necessary in this protease. Number C, aspartate is the most important in the catalytic triad. And number D, histidine is sufficient to activate these proteases because of the more nucleophilicity of sulfur. So read the options carefully and let me know in the chat box which of the following you think is the correct answer. Again I am telling you, it doesn't matter if your answer is wrong, but at least you should try. Read the options very carefully and then try to answer the question. So, is there anybody tried to answer the question? Number A, number A, number A. So, number A is the most given answer today for this question. So, let me check. So, the correct answer is number D. Histidine is sufficient to activate these proteases because of the more nucleophilicity of sulfur. Let me show you what is the active site mechanism for the thiol proteases. So, as you can see, in thiol proteases there is a catalytic triad, but not necessarily the catalytic triad will be for aspartate, histidine and cysteine only. Okay. The aspartate, it is not the most important moiety for this thiol protease. It may be other amino acids also, but histidine and cysteine, these both are very important part for the thiol proteases. Okay. As the name suggests, thiol means there is a SH group, right? So, thiol is nothing but this SH group, and we all know which amino acid contains this thiol moiety is the cysteine. Okay. The cysteine is present in the catalytic triad, histidine also. Here histidine has this imidazole kind of imidazole side ring present in the histidine. Okay. So, the sulfur nucleophilicity is very high. That is why this imidazole group of histidine is sufficient to activate this protease So, what happens here? Just like I have shown you in the aspartate protease, here the histidine nitrogen lone pair, it abstracts the proton for this thiol moiety. S negative charge is there, and this negative sulfur, it attacks the carbonyl carbon here. Okay. And this kind of tetrahedral intermediate is formed. And now this what happens, this oxygen negative charge again makes this double bond, and the bond hydrolysis happens here. This RNH takes a proton from the histidine moiety, and we have this kind of structures. Again water molecule comes and they actually breaks this bond, carbonyl carbon and sulfur bond, and we have this hydrolyzed product. Okay. So, let us again check what were the options. It said that it can only react via the catalytic triad of aspartate, histidine and cysteine. It is not true, because cysteine and histidines are important, but aspartate is not that much important. So, there may be another amino acid in place of aspartate. Okay. It is not constantly present in all thiol protease. Histidine is not necessary. It is also incorrect, because as we have seen from this active site mechanism, histidine is extremely necessary for the thiol proteases. Aspartate is the most important in this catalytic triad. It is also not true, but histidine is sufficient to activate these proteases because of the more nucleophilicity of sulfur. It is the true. Okay. So, this was about your problem number 3. So, now I will move toward the next problem. Now, problem number 4, it says that which inhibition study deals with the decrease of the value of both Vmax and Km with the increase in the inhibitor concentration. Your options are number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, and number D, suicide inhibition. So, which inhibition study deals with the decrease in the value of both Vmax and Km with the increase in the inhibitor concentration? I will show you few graphs in for this question, and we will discuss each and every inhibition later in today's class itself. What is a competitive, what is a non-competitive, what is uncompetitive, what is a suicide inhibition, all of these inhibitions we will discuss later in today's session itself. This is a more general question regarding these inhibitions. I have discussed what is a Vmax and what is a Km or Michaelis-Menten constant in last class. So, Vmax, what is Vmax? Vmax is the maximum the reaction rate, okay, and the Km or Michaelis-Menten constant is the substrate concentration at the half Vmax. And this question is related to a particular graph for the enzyme inhibition. I have discussed that graph also in last day class. So, try to answer this question. Which of the following you think is the correct option? Which inhibition study deals with the decrease of the value of both Vmax and Km with the increase in the inhibitor concentration? Is it competitive, is it non-competitive, is it uncompetitive, or is it suicide inhibition? So, let me check if there is any answer. Okay, so I have few answers for this question, I think. Number A, number B, number A, number A, B. Okay, I think number A and number B, most of the answers are related to this. Okay, let us check what is the correct answer. So, the correct answer is number C, uncompetitive inhibition. Now, this is the mechanism for the, or this is the Lineweaver-Burke plots of the mechanism of enzyme inhibition. Okay. So, as you can see, look at carefully for the graphs. Okay, the particular graphs are there. Okay. So, this is the first graph, it is for the competitive inhibition. Okay. So, what happens here in the graph? So, as we all know, Lineweaver-Burke plot, it is between what? It is between in the y axis. In the y axis we have 1 by V, okay, and in the x axis we have 1 by concentration of S, means 1 by concentration of substrate, okay, and the graph actually look like this. So, the red curve, it is for the in presence of inhibitor, and the blue curve, it is for the no inhibitor. Okay. So, what happens, as we all know, the intersection, what is the intersection? For this graph, the intersection is 1 by Vmax. Okay. The intercept is 1 by Vmax and the slope is the Km by Vmax. So, if we actually Lineweaver-Burke plot, it is a straight line plot, and if we just compare it with the y equal to mx plus c. So, the c is the intercept, which is the 1 by Vmax, and m is the slope, which is Km by Vmax. Now, look at carefully to the each and every graph. So, for the first one, let us discuss everything. Here in the competitive inhibition, it is said that Km is increased, Vmax is unaffected. So, if you look at carefully, when there is a no inhibitor, the graph looks like this. When there is a inhibitor, the graph looks like this. So, in both cases, what happens? The both curve, they actually intersect in a same point in the y axis here in this point. So, in this point, the both graphs are intersecting. So, when the both graphs are intersecting in the both straight lines are intersecting in the same point on the y axis, what does it mean? The value of intercept is the same, whether there is an inhibitor or not, which means the Vmax is constant here. Okay, the Vmax value, 1 by Vmax value is constant here. If 1 by Vmax is constant, means Vmax is constant. Okay, but as you can see from this curve, when there is an inhibitor present, it is much more steeper than this curve. Okay, what does it mean is if it is much more steeper, it means that the value of the slope is higher. So if the value of the slope is higher here, so if the value of the slope is higher for in case of presence of inhibitor, means what does it mean? What is the value of the slope? The slope is this, Km by Vmax. Okay. So if in presence of the inhibitor the value of the Vmax is constant, but the slope is increasing, which does mean that the value of Km by Vmax is increasing, means the value of Km is increased. So it was for the first case, means for the competitive inhibition. Now we will come to in the uncompetitive inhibition. So what is happens here? Look at the straight lines now here. So the first red curve, it is for the in presence of inhibitor, and the blue straight line is for the no inhibitor. Okay, here as we can see there is a substantial difference between the intercepts. Okay, so as you so for in presence of inhibitor, what it is happening, the amount of intercept it is increasing. This intercept value is more than this intercept value. So if let us say in presence of, so 1 by Vmax will be increasing. So it is greater than 1 by Vmax. Let us say it is 1 by V1 and it is 1 by Vmax 2. So if let me just use the eraser here. So if what happens, if 1 by Vmax value in presence of inhibitor, now if I am adding the inhibitor, the 1 by Vmax value is increasing. Now if this 1 by Vmax value is increasing, what does it mean? It means Vmax value is decreasing or Vmax value is reduced, which is mentioned here. Now as you can see, the slope is same. Both of the lines, they have same slope. Okay, so if the slope is same, so what is the slope? Again, slope is the Km by Vmax. If the slope is same but the Vmax value is decreasing, what does it mean? Means to make the slope constant, Km also needs to be decreased. Okay, so for example, if the slope is, let me say the slope is 1 by 9. Let me okay, let me erase it. Okay, let me say that the slope of this particular graph was 4 by 9. Okay, now if you add the inhibitor, so the value of Vmax is 9 and the value of Km is 4. Now if what happens when you are adding inhibitor, what is happening? The value of Vmax is reduced. So let us say the value of Vmax becomes 3. So if the value of Vmax becomes 3, to keep this ratio constant, the value of Km also need to be reduced to 4, so that the slope remains constant as in 2 by 3. So that's how it works. Okay, if you remember how the curves, the straight lines look like in presence of inhibitor and without inhibitor, the Lineweaver-Burke plots, then you can easily answer this kind of question. Now look at for the particular it is the non-competitive inhibitor. Now in case of non-competitive inhibitor, what happens? The Vmax value again it is getting reduced. Okay, here Vmax value is getting reduced, but here also what happens, when the Vmax value is getting reduced, there is a difference of slope, and if you extrapolate these both lines actually they join together in the negative x-axis. Okay, since these both of the straight lines have the same value of minus 1 by Km, what does it mean? The Km is constant in case of this non-competitive inhibition. Okay, so it was the example for the non-competitive inhibition. So for this kind of inhibition, what you need to remember is, what is the particular inhibition, what are the definitions of the particular inhibition, and what does each and every inhibition mean, what is the Lineweaver-Burke plots for the each and every inhibition. Okay, and how the value of Km and Vmax is changing. If you can remember the particular graph, how the particular straight line graph is looks like, you can easily answer this kind of question. Okay. So look at the graphs carefully when I will show you this PPT. Read about the graphs carefully and try to write on your copy about the graph. Take each and different values for the Km and Vmax, then try to solve this kind of question. Okay, it will be, I think, easier for you. So now I will move toward the next problem. So problem number 5, it says that which one of the following inhibition is called the mechanism based enzyme inhibition. Okay, your options are number A, competitive inhibition, option B, non-competitive inhibition, option C, suicide inhibition, and option D, active site directed irreversible inhibition. Which of the following is called the mechanism based enzyme inhibition? Try to answer this question. Even if your answer is wrong, please keep trying. I will again show you the problem number 5. Which of the following inhibition is called mechanism based enzyme inhibition? Is it competitive inhibition? Is it non-competitive inhibition? Is it suicide inhibition? Or is it active site directed irreversible inhibition? So, have anybody tried? Okay, I have few answers like number C, number D, number C, number D. Okay, so I think there are equal number C and number D. Okay, let us check. So the correct answer is number C, suicide inhibition. So let us discuss what is suicide inhibition. So suicide inhibition, it is also known as mechanism based inactivation. Okay, the inhibitor makes the use of enzyme's own reaction mechanism to inactivate it. In suicide inhibition, the structural analog is converted to a more effective inhibitor with the help of the enzyme to be inhibited. For example, ornithine decarboxylase, or ODC, it catalyzes the conversion of the ornithine to putrescine, and which is necessary for the polyamine synthesis. When the ODC in trypsinoma is inhibited by multiplication of the parasite is arrested. Therefore, inhibitors of ODC enzymes such as difluoromethyl-ornithine or DFMO have been found to be effective against the trypsinomyces or sleeping sickness. A similar mechanism is observed in case of allopurin, which is oxidized by xanthine oxidase to alloxanthine, that is a strong inhibitor of the xanthine oxidase. The anti-inflammatory action of aspirin is also based on the suicide inhibition. The arachidonic acid is converted to the prostaglandin by the enzyme cyclooxygenase. Aspirin acylates the cysteine residue in the active center or cyclooxygenase. Thus, prostaglandin synthesis is inhibited. So, inflammation subsides. So, enzyme, what happens generally in suicide inhibition? So, the inhibition, as we all know that every enzyme has its own mechanism to work on the substrate. Okay. So, what happens in the suicide inhibition, the particular inhibitor molecule, they actually reacts with the substrate. okay, the enzyme molecule, and what it makes a more effective inhibitor, okay, with the help of the enzyme which is inhibited. So actually it uses the enzyme's own power or enzyme's own mechanism to particularly inhibit it. So this is basically kind of a suicide, like the enzyme is killing its activity by its own. That is why it is called the suicide inhibition, and more it is called the mechanism based inactivation or mechanism based enzyme inhibition. Okay. So now it was the problem number 5 and there were some correct answers. So now I will discuss the next problem. Problem number 6, it says that which of the following is not true of the suicide substrates. Option A, they are activated by the enzyme catalyzed mechanism reaction. Number B, they react irreversibly with the enzyme. Number C, they form a covalent bond with the active site once activated. Number D, they self-destruct as a result of an enzyme catalyzed reaction. So read the options carefully and try to answer this question, which of the following is not true of suicide substrates. Number A, they are activated by the enzyme catalyzed reaction mechanism. Number B, they react irreversibly with the enzyme. Number C, they form a covalent bond with the active site once activated. Number D, they self-destruct as a result of an enzyme catalyzed reaction. Try to answer this question in the chat box. Please try it. It doesn't matter if it is correct or wrong. Which of the following is not true of suicide substrates? Option A, they are activated by the enzyme catalyzed reaction mechanism. Option B, they react irreversibly with the enzyme. Option C, they form a covalent bond with the active site once activated. Option D, they self-destruct as a result of an enzyme catalyzed reaction. So is there any answer for this question? Okay, so number C, number C, number A, number. So I have 2 number C and 2 number A. Let us check which of the following option is not correct. It is number D. They self-destruct as a result of an enzyme catalyzed reaction. Let me check the answer. So what are the suicide inhibitors or suicide substrate? Inhibitor binds as a substrate and is initially proceed by normal catalytic mechanism. Okay, then it generates a chemically reactive intermediate that inactivates the enzyme through the covalent modification. Suicide because enzyme participates in its own irreversible inhibition. Okay, so what is a suicide inhibition? It is a special type of irreversible inhibition of the enzyme activity. It is also known as the mechanism based inactivation. The inhibitor makes use of enzyme's own reaction mechanism to inactivate it. Okay. In suicide inhibition, the structural analog is converted to an more effective inhibitor with the help of the enzyme to be inhibited. The substrate like compound initially bind with the enzyme and the first few step pathway are catalyzed. This new product irreversibly bind with the enzyme and inhibits the further reactions. Okay. So the suicide substrate, they itself, they do not actually destruct the particular reaction mechanism, the new product which is formed, they actually irreversibly binds to the enzyme and then inhibits the further reaction. Okay, so what let us check the options more carefully now. Option A, it says they are activated by the enzyme catalyst reaction mechanism, which is true because we all have this seen in the last slide also. It is a special type of mechanism which makes use enzymes own reaction mechanism to inactivate it. Okay, they react irreversibly with enzyme, it is also correct. Okay, they actually irreversibly irreversibly participate with the enzyme inactivation. They form a covalent bond. So, whenever there is a irreversible active binding with the enzyme, okay, the covalent bond formation will happen because of the irreversible activity, because once a covalent bond form, there is a irreversibility comes, because in the breakage of the covalent bond the things changes for the enzyme anatomy. They self destruct as a result of enzyme catalyzed reaction. This statement is not true because they not itself, they do not destruct the enzyme. Okay, they actually bind with the enzyme. The new product is from that product that destructs the enzymes particular enzymes mechanism. Okay, so this question was a little bit tricky, but if you read in more detail, you will be, I think you will be understand what is the correct statement. Okay. So, now I will move toward the next problem. Problem number 7, it says that the active site of chymotrypsin consists of a catalytic triad of which of the following amino acid residue? Your options are: number A serine, histidine aspartate, number B serine, histidine glutamate, number C threonine, histidine aspartate, and number D methionine, histidine glutamate. What is the catalytic triad which is present in the chymotrypsin enzyme? Serine, histidine aspartate, serine, histidine glutamate, threonine, histidine aspartate, methionine, histidine glutamate. Try to answer this question in the chat box. The active site of chymotrypsin consists of which amino acid residues? Let me check. Number A, number A, number A. So, number A is the most frequently given answer. So, the correct answer is yes, very good. It is number A, serine, histidine and aspartate. So, the catalytic triad, 3 amino acid residues in the active site in a hydrogen bonded network. Okay, it is called the catalytic triad. So, in case of chymotrypsin, the amino acids are present, is serine, the residue number 195, histidine, the residue number 557 and aspartate, the residue number 102. This is essential for the effective catalytic activity in chymotrypsin. Catalytic triad action converts OH group of serine into a potent nucleophile. So, what is the role of this catalytic triad is basically making the serine moiety, the hydroxyl group of serine to lose the proton and to make it in the alkoxide ion or O minus ion. And how it happens? This aspartate has a negative charge, this carboxylate acid, right, and it actually attracts the proton from this histidine imidazole moiety, and in turn it makes the histidine moiety positive, and then this positive histidine, it is this nitrogen becomes more nucleophilic, the lone pair of nitrogen, it attracts the proton for this hydroxyl group of the serine and makes it the alkoxide ion O minus, which is a better nucleophile. So the residue numbers are nothing but when there is a polypeptide chain, the amino acid which is in the N terminus, it is numbered as 1, and the amino acid is the at the last C terminus, it is numbered as the whatever the number. So in a polypeptide chain there are many amino acids are there. So here in the chymotrypsin enzyme, the residue number 57 is the histidine, residue number 102 is the aspartate, and the residue number 195 is the serine. Okay. The tetrahedral intermediate of the serine protease mechanism is this. This is the particular protease. So this is the NH of the protein 1 prime, and this is the peptide bonds. This is the another polypeptide side. Okay. The aspartate 102 stabilizes the positive charge acquired by the histidine 57. The negative charge on the carbonyl oxygen of the protein 1 residue is the substrate is stabilized by the main chain amides of the enzyme that comprise the oxyanion hole. So this is the oxyanion hole, okay, of the tetrahedral intermediate. So it was a correct. So number A was the correct answer. Now I will move toward the next problem. So which of the following statement is false regarding ASDII? Number A, TPCK is an inhibitor of chymotrypsin. Number B, TLCK is an inhibitor of the trypsin. Number C, the inhibitors do not have any directional property. And number D, the reactive functionality of the inhibitor is responsible for this kind of mechanism. So try to answer this question. So what is ASDII? It is nothing but a active site directed irreversible inhibition. Okay. Now read the options carefully and try to answer which of the following is the correct answer. I will show you the correct answer shortly with the what is the ASDII. But before that you have to try to answer it. So let me check the chat box. So I have few answers, number D, number C, number A. So I have few answers like number D, C, A. So we have multiple answers here. Let us check what is the correct answer. So the correct answer is number C. So I think we have number C in the chat box. So very good. So let us discuss now. Active site mapping via irreversible inhibitor. So what is the ASDII? What is the full form of it? It is active site directed irreversible inhibition. The name itself suggests that these inhibitors must have a particular directional property. That is why option number C, it is incorrect. Okay. And the TPCK, it is a particular compound which is the inhibitor of chymotrypsin. Okay, and here it is the full form of TPCK, it is the tosyl L-phenylalanine chloromethyl ketone. It is an inhibitor of the chymotrypsin, and TLCK, it is an inhibitor of the trypsin. And the reactive functionality of inhibitor, it is responsible for this kind of mechanism, it is also correct. So now what is the mechanism? So active site mapping via irreversible inhibitors. So, this is the neutral substrate for the chymotrypsin, this kind of substrate. So, we all know how the chymotrypsin reacts. Basically, there must be an aromatic group or aromatic amino acid. Okay. So, this is the normal natural substrate. Now, this substrate, it is the inhibitor moiety and it mimics the particular natural substrate for the chymotrypsin, and it has a particular reactive group here. Okay. Now, this if the chymotrypsin, we have this histidine 57 moiety, which is very important in the catalytic triad, just we have seen in the last slide. Okay. This histidine 57, as I have shown you in the last slide here, the histidine 57 is a very important part for the catalytic triad of chymotrypsin. Now, this TPCK, it reacts with this particular histidine 57 and it makes it inactive by reacting with this reactive group of this TPCK inhibitor, and it is the example of this active site direct irreversible inhibition, where the active site of the particular enzyme is directing the irreversible inhibition of the same. Okay. So, now option number C was incorrect. So, very good. Now, we will move toward the next problem. Problem number 9, it says that which of the following is known as antigenic determinant? Your options are number A hapten, number B epitope, number C abebzyme and number D paratope. So, which of the following is known as antigenic determinant? Try to answer this in the chat box. After that, I will discuss in detail what is a hapten, what is a epitope, what is abzymes, what is paratope. Everything I will discuss in detail. So, which of the following is known as an antigenic determinant? Is there any answer in the chat box? Number A, number B, number B. So, number B seems to be the most frequently given answer for this question. Now, let me check if it is correct or not. So, the correct answer is very good. Number B, epitope, is known as an antigenic determinant. Now, let us discuss what is a hapten, what is epitope, what is antigenic determinant, what is abzymes and what is paratope. So, basically haptens, a hapten is a molecule too small to stimulate antibody formation by itself. However, when the hapten is combined with a larger carrier molecule, usually a serum protein, the hapten and its carrier together form a conjugate that can stimulate an immune response. So, these small molecules, they are known as hapten, and when it is combined with a carrier molecule like a big larger molecule of like serum protein, it will make a hapten carrier conjugate, and this conjugate can stimulate the immune response. Okay. What are abzymes? Abzymes this is a combination from an antibody and enzyme. It is often called a catalytic antibody and it is a monoclonal antibody with a catalytic activity. So, this is a abzyme, as you can see the formation. This is a abzyme domain, this is a antibody domain. Okay. And here the inactive prodrug, they became an active drug by the prodrug activation, and here the tumor or cancer cell binds. Okay. So, here the cancer cell or tumor binds in this antibody domain, and here in the catalytic domain the prodrug or drug molecule bind, and that is how this abzyme formation it happens. So what is a epitope and what is a paratope? An epitope is known as antigenic determinant. It is a part of an antigen that is recognized by immune system, specifically antibodies, B cell, T or T cells. Paratope is a part of an antibody which recognize antigen binding site of an antibody, and it is a small region of 15 to 22 amino acids of the antibodies region, and that contains the antibodies heavy and light chain. So as you can see, this is the antibody, okay, and this is a particular space where the antigen molecule binds, and here it is called the paratope. As you can see, there is a particular curve here, and this in this curvy region, this curves of the antigen molecule fits. So it is a paratope, and this is the antigen where there is a particular because part of the enzyme, it is called the epitope, which can be recognized by the antibody. So remember, epitope is a part of an antigen, and that's it is called antigenic determinant. Paratope is a part of the antibody which can bind with the epitope, or the which can bind with the antigen. Okay. So this was your problem number 9. Moving forward to the problem number 10, it says, where does inhibitor binds on enzyme in a mixed inhibition? Your options are number A, allosteric site, option B, active site, option C, binds on the substrate, and option D, doesn't bind on enzyme. So where does an inhibitor molecule binds on an enzyme in a mixed inhibition? So as I have mentioned, there are many kind of inhibition. There is a competitive, incompetitive, and mixed inhibition is one of them. So in this mixed inhibition, the particular inhibitor molecule binds to the enzyme to a particular position. What is the position? Allosteric site, active site. It binds on the substrate itself, and it does not bind on the enzyme. Identify the correct answer. So is there any answer for this question? Okay, number A, number B, number B. So I have 2 number A and 2 number B. Let me check. So the correct answer is number A, allosteric site. Okay. So let us check. So what is the difference between an allosteric site and active site? An allosteric site is a region of an enzyme where activator or inhibitor molecules can bind either to activate or decrease the enzyme activity, and the active site of an enzyme is the area where substrate molecules bind and the enzyme catalyzes the reaction, which results in the creation of the specific products. It is only found in allosteric enzymes. It founds in all enzymes. The active site founds in all enzymes. Allosteric site, it's actually on substrate, and it is the effector molecule. Do not have 2 separate web site. Binding and catalytic sites are 2 different sites. In non-competitive or mixed inhibition, what happens? There is an enzyme and there is a 2 side. One is the active site and one is the allosteric site. Here one is the active site, this V shaped. Okay, sorry, there is a mistake by my side. Okay. So here it is the active site, this V shape, and this semicircular, it is the allosteric site. As you can see, there is a substrate and there is a inhibitor. So now when the first the inhibitor molecule binds with the substrate and the active site conformation changes. and enzyme active inhibitor complex, the active site is changed. So, inhibitor binds to the allosteric site and changes the shape of the active site, and now the particular molecule of the substrate, it cannot bind with the enzyme and the reaction does not happen. So, in the mixed or non-competitive inhibition, what happens, there is an enzyme, okay, this free enzyme, it have inhibitor binding site which is an allosteric site and a substrate binding site. It is an inhibitor molecule and it is a substrate molecule. So, when there is no inhibitor present, the substrate molecule will bind with particular enzyme, the enzyme substrate complex will form and products will form. But when there is a inhibitor molecule when it binds with the particular enzyme, the substrate molecule cannot bind with the enzyme anymore, and what will happen, there will be no enzyme catalyzed reaction here. Okay. So, that is how the mixed inhibition on the non-competitive inhibition happens. So, now I will move toward the next problem. Okay. So, I will read you the problem number 10. You have to read it carefully and then write the answer. Okay. Meanwhile, I will give you a 2 minutes break. After answering, I will not I will keep my camera and voice off for 2 minutes, but do not leave the class. Try to answer the problem number 10 in meanwhile. Okay. So, it says a student is conducting an experiment in which he adds an inhibitor to an enzyme catalyzed reaction. When the student first adds the inhibitor, the reaction rate decreases. However, he can return the reaction rate to normal by adding large quantity of substrate. What type of inhibitor is he using? A non-competitive inhibitor, and a competitive inhibitor, an uncompetitive inhibitor. The student likely made a mistake, the result unacceptable regardless of any inhibition. So, I am keeping my camera off for 2 minutes, okay, only 2 minutes I am giving you for giving the question in the chat box, okay. This does not matter if it is correct or incorrect, but read the question very carefully. So, I am keeping my camera and voice off for 2 minutes, but I am not leaving the class, okay. I am giving you just 2 minutes, I will check the answers after 2 minutes. So, try to answer this question. Hello. Okay, so I have rejoined. So can you hear my voice and can you see me? Please somebody confirm if my voice is clear. Yeah. So thank you very much. Now I have two answers and it is saying number B. Okay, let me check what is the correct answer. So the correct answer is very good. It is number B, competitive inhibitor. So competitive inhibition, enzymes can bind with the substrate forming enzyme substrate complex or inhibitor but not both. Okay. Competitive inhibitor resembles the substrate. Competitive inhibition can be relieved by increasing the substrate concentration. So what does it mean? What is the competitive inhibitor? So competitive inhibitor is a such molecule which resembles just like a substrate molecule. So your enzyme cannot differentiate between a competitive inhibitor and the substrate. So when if you start a particular reaction and you add a competitive inhibitor, the enzyme molecule will think it is a substrate and it will bind with the particular inhibitor. Okay, here the enzyme can bind only either to the substrate or to the inhibitor. Now when you are adding the inhibitor, the reaction rate will obviously decrease. Okay, because some inhibitor enzyme molecule will be binding with the competitive inhibitor, but after some time if you increase the amount of the substrate, now what will happen? If the substrate is in excess, the enzyme substrate concentration will also enzyme substrate complex concentration will also increase and the reaction rate will return to the normalcy. So what happens in the competitive inhibition? Here is the enzyme and here is a substrate. Okay, and here is a inhibitor. Now if inhibitor binds and the substrate inhibitor looks like very same, means their reactivities or their various functionalities are similar. So if they both are binds, the substrate molecule cannot bind with the enzyme and product will not form. Okay, the graph of the particular competitive inhibition is like this, as I have shown. Okay, here but if what happens, so this is the graph, as you can see, as I mentioned, the Km value in presence of inhibitor, the Km value will increase. Okay, and the Km, so this is the particular value, Vmax value will remain same. Okay, but the slope of in presence of inhibitor, the slope increases, so Km value increases, as you can see from this graph also. The blue one is without any inhibitor when the red one is with inhibitor. Okay, so if you increase the substrate concentration, the rate will become similar. This is for the competitive inhibition. Now problem number 11, it is a mathematical problem and I want all of you to try to answer this question. Just take a pen and paper, it is very easy mathematics, and use the Michaelis-Menten kinetics. Okay, so it says that consider the following plot of the velocity versus substrate concentration of the enzyme catalyzed reaction obeying Michaelis-Menten kinetics with an initial enzyme concentration of 10 to the power minus 5 micromolar. The turnover number of the reaction is 8000, this is 8 lakh, this is 40,000 and this is 4 lakh. This is the graph which you have to use. There is a reaction rate in the y axis and the substrate concentration in the molar 10 to the power 3 molar concentration. Okay. So what is the turnover number of the reaction? Try to find out, try to solve this problem. So have anybody tried? Take one more minute, then I will show you how to solve this kind of problem. Just look at the graph carefully. Here you have to use the values generated from the graph. Take some time. These kind of mathematical problems are very important to you for your exam purpose and also for your understanding of the course. So I have one answer, number A. Okay, let me check. Okay, take one more minute and then I will show you the answer. Let us check the answer. So the correct answer is number B. It is 8 lakh. Okay. How to solve this kind of question? Now let us check. So what is the laser pointer? Yeah. So Michaelis-Menten equation, what is it? We all know V0 equal to K2 S0 E0 by S0 or Km. So as we all know what is the Michaelis-Menten equation. Okay, this K2 S0 and then at high substrate concentration what will happen in the high substrate concentration. So this equation I have shown you in the last day also, right? Only thing is which is different is the K2 E0 is altogether written as a Vmax, which is written here like this. Okay, so Vmax can also be written as K2 E0. Okay, somebody wants to join. So as I have said, Vmax can also be written as the K2 E0, where the K2 is the turnover number, okay, and E0 is the initial enzyme concentration. Okay, so it this Vmax is also known as the rate attains at the maximum value and the reaction is 0 order with respect to substrate concentration. So what will happen? This is the initial equation. If at the higher substrate concentration, when the substrate concentration is very much greater than Km, so in the denominator we can neglect the Km with respect to the substrate concentration, and the equation will become like, if we ignore Km, then the equation will be V0 equal to K2 S0 E0 divided by S0. S0 cancels out and it will be V0 or rate equal to K2 E0 or equal to Vmax. This K2 is called the turnover number of the enzyme, that represents the number of molecule converted to the product in a unit time by one molecule of enzyme. So remember this thing. This K2 is the turnover number for the enzyme. It represents the number of the molecules converted to product by one molecule of the enzyme. Okay. So according to the question, now what is the question? The question says that the initial enzyme concentration is 10 to the power minus 5 micromolar. So E0 is 10 to the power minus 5 micromolar and the Vmax is 8 micromole per minute. So from where I got the Vmax? From this graph. Now look at the graph carefully. If you extrapolate this graph, okay, so it crosses at 8 and the unit is micromole per minute. So Vmax will be 8 micromole per minute. Now K2 will be equal to Vmax by E0. So if you do the Vmax by E0, you will get this answer. Micromole will be cancelled out. It will be 8 into 10 to the power 5. Okay, 8 into 10 to the power 5 minute inverse. So your correct answer is number B. So that's how this kind of problems are solved. So remember few things from here, like what is the turnover number? Turnover number of a enzyme represents the number of molecule converted to product by unit time by one molecule of enzyme. The rate of the reaction, the maximum rate attains in a enzyme catalyzed reaction, Vmax equal to K2 or turnover number of the enzyme into initial enzyme concentration, which is E0. Okay. Now, problem number 12, it says that consider the following plot of 1 by V0 versus 1 by S plot of Lineweaver-Burke. The result obtained from the graph is intercept value is 0.1 micromolar per minute inverse, and the slope is 9 into 10 to the power 5 minute. What is the value of Km? Your options are 9 into 10 to the power 5 micromolar, option B 0.11 into 10 to the power 5 micromolar, option C 9 into 10 to the power 6 micromolar, and option D 0.011 micromolar. Now, all of you know what is the Lineweaver-Burke plot, what is in the x axis, what is in the y axis, and what is the value of slope and what is the value of intercept. I have mentioned it before in today's session, in last session also. Just put all the values together and you will find out what is the Km value of the reaction. Take 2 minutes and try to solve this answer question. So, is there any answer for this question? Let me check. Okay, till now it is not there. Okay, let me give you 2 more minutes, 1 more minute, and then we will discuss it. Okay. Anyway, this kind of problems are very common from the enzyme catalysis or the particular enzyme catalysis. This kind of mathematical problems are very common. So, I will I will suggest that all of you just practice all the graphs very carefully, what is in the x axis, what is in the y axis, and what are the slope intercept values, because from these you can expect few mathematical questions. Okay. Let me show you the answer first. So, correct answer of this question is number C, 9 into 10 to the power 6. Okay. Now, let us solve it. So, this is the Lineweaver-Burke plot. I have shown you earlier this plot frequently in today's class, in last class also I have shown you this plot. Okay. So, what is in the Lineweaver-Burke plot? In the y axis we have 1 by V, in the x axis we have 1 by S. This is the graph, it is a straight line, the intercept value is 1 by V max and the slope is Km by V max. Okay. And the negative intercept value or negative x axis intercept value is minus 1 by Km. Now, from this maths, what is it is said? It says that the Lineweaver-Burke plot is associated with the equation 1 by V0 equal to 1 by V max plus Km by V max into 1 by S. Now, if you compare this particular equation, if you compare it with the y equal to normal straight line equation, y equal to C plus mx. So, here you can see the C is the value of the intercept, which is this 1 by V max is the value of the intercept. The value of slope or m is the Km by V max, which is here. So, now from the equation it is said that 1 by V max is 0.1 micromolar minute inverse. Okay, so the inverse will be only on this, the value is not on the inverse, and the V max value is 10 So, what is the Vmax value? The Vmax value is 10 micromolar per minute. Now, the slope or Km is Km by Vmax. So, the Km value will be slope into Vmax. The slope is 9 into 10 to the power 5 minute and the Vmax is 10 micromolar per minute. So, if you multiply it, you will get the value of Km, which is 9 into 10 to the power 6 micromolar. Okay. So, that is how you have to solve this kind of problems. Okay, it was problem number 12. Now, similar I have one more problem, mathematical problem for you. Now, I want all of you to answer the next question, because it is very similar to this problem and I want all of you to try to answer that problem. So, it says that consider the following plot of the S concentration S by V0 versus S plot of the Hanoi wolf. Okay, Hanoi wolf plot is similar to the Lineweaver-Burke plot. For here in the y axis we have concentration of S by V and in the x axis we have concentration of S, and this is the straight line. So, the results obtained from the graph is intercept value is 0.5. Okay, the slope is 0.0002 millimolar inverse. Now, what is the value of Km of the reaction? Number A 5000 millimolar, number B 500 millimolar, number C 2500 millimolar and number D 250 millimolar. Now, this is a very easy, because I have solved the last problem, and if you have paid attention you can solve it very easily. Just see what is the intercept, what is the particular slope, the values are given and just you have to calculate the Km value. So, take 2 minutes and I want all of you to answer this question. If you have understood the previous problem, how we solved it, you can solve this question also. Since Hanoi wolf plot is not as much I have discussed, because it is not so much common like Michaelis-Menten plot and the Lineweaver-Burke plot. So, I have given the plot itself here. Now, just put the values and let me know what is the correct answer. I am giving you 2 minutes. All of you please try to solve this question. It's just like the previous one, just put the values together and you will have the answer. Let me check if I have any correct answers. Okay, I have quite a few answers, maybe B, A, D, B. So, I have 2 number B and 1 number D and 1 number A. Okay, so let me check. So, the correct answer is number C. Okay, so how? Just you have to put the answers here again. The Hanoi-Wolf plot is associated with the following equation. It is S by V0, which is in the y axis, equal to S by V max. Okay, it is your n plus Km by V max. So, Km by V max is the here it is the intercept value. As you can see from the graph itself, the Km by V max is the intercept and S is the x. Okay, so 1 by V max will be your slope. Now, in the exam question, it is saying that intercept value is Km by V max, slope is 1 by V max. Now, from the question, Km by V max is 0.5, right? It is the intercept, and 1 by V max, the slope is 0.0002 millimolar inverse. So, V max is 5, if you just revert it, so you will get 5000 millimolar. Now, just you have to multiply the Vmax, okay, the Vmax with this value Km by Vmax is 0.5. So, what is the Km? Km will be 0.5 into Vmax, means 2500 millimolar. See, this kind of mathematics, it is not hard at all. These are very easy because you are given all the values. You just have to put the exact value for the particular thing. Like if you are given the value of the slope and intercept, you just have to, the slope is 1 by Vmax. So, you have to put 1 by Vmax equal to this. You have to put the value of intercept Km by Vmax is 0.5. From that thing, from 1 by Vmax you can calculate Vmax. Now, from Km by Vmax, if Vmax is known, you can easily determine the Km. That is how this kind of problems are solved. Okay. If you practice more, I will suggest please practice more of this kind of problems. Okay. Just then you will find it very easy to solve any problems related to this kind of plots. Now, next problem, it says that which of the following statement is incorrect. Option A, production of monoclonal antibody is expensive. Option B, polyclonal antibodies are produced by same clone of plasma B cell. Option C, production of polyclonal antibodies does not require hybridoma cell lines. Option D, monoclonal antibodies are used as therapeutic drugs. So, you have to identify which of the following statement is incorrect regarding the monoclonal and the polyclonal antibodies. Try to answer this in the chat box. So, let me check if there are any correct answer for this. So, you do not have to know because it is a, I believe this all of these things were in your week 4 class session. If you have paid attention to this class, I think you will know, but anyways, number C is more given, frequently given answer. So, let me check. So, the correct option, or the rather incorrect option is number B. Production of polyclonal antibodies are produced by same clone of plasma B cells. It is the incorrect statement. Now, let us discuss what is polyclonal and monoclonal antibodies. So, monoclonal antibodies are the antibodies that are identical because they were produced by one type of immune B cell, all clones of a single parent cell, and polyclonal antibodies represent the antibodies from multiple clones, okay, of the B lymphocytes, and therefore they bind to a number of different epitopes. For example, 4 immunoglobulin. So, these are the differences between the monoclonal antibodies and polyclonal antibodies. So, the first one, monoclonal antibodies, they refer to a homogeneous population of antibodies that are produced by a single clone of plasma B cell, but polyclonal antibodies refer to a mixture of the immunoglobulin molecules that are secreted against a particular antigen. The first one, they are produced by the same clone of plasma B cell. Polyclonal antibodies produced by different clones of plasma B cell. Monoclonal antibodies production require hybridoma cell lines, but the polyclonal one, their production does not require hybridoma cell line. A homogeneous antibody population is the monoclonal one, the polyclonal one is the heterogeneous antibody population. The first one interact with a particular epitope of antigen, and the second one, they interact with the different epitopes of the same antigen. The production of the monoclonal antibody is expensive, but the production of the polyclonal antibody is not expensive. Skills are required to handle the technology for the monoclonal antibody. Fewer skills are required for the production of the polyclonal antibodies. The production of monoclonal antibodies takes some time. Production of the polyclonal antibody takes less time. They possesses less cross reactivity. They possesses comparatively high cross reactivity. They are used as therapeutic drugs. They are used as a general application. Advantages include immortal supply, high specificity and high reproducibility. Advantages include high affinity, tolerance of minor changes and more robust detection. Okay. So, these were the difference of the polyclonal and monoclonal antibody. Okay. So, the statements number C, the statement number B is incorrect. They are not produced by the same clone of plasma B cell, rather they are produced by different clones of plasma B cells. Okay. Now, problem number 16, what is the use of the hybridoma technology? Number A, separation and isolation of monoclonal antibody from polyclonal state. Number B, separation and isolation of lymphocytes from immunoglobulins. Number C, separation and isolation of haptens from epitopes. Number D, separation and isolation of haptens from enzymes. So, what is the use of the hybridoma technology? Try to answer it in the chat box. After you try to answer, we will discuss what is hybridoma technology and what is its use. So, let me check which is the correct answer and is there any answer in the chat box. Okay, so number A, number D, number C. So, I think there are different answers till now. Okay, let us check what is the correct answer. So, the correct answer is number A, separation and isolation of monoclonal antibody from polyclonal set. Okay, so hybridoma technology is a method for producing large number of identical antibodies called monoclonal antibodies. It was discovered by G. Kohler and C. Malmsten in 1975. They were awarded Nobel Prize for Physiology and Medicine in 1975. The hybrid cells are produced by fusing B lymphocyte with myeloma cells or tumor cells. The B lymphocytes have the ability to produce large number of antibodies and tumor cells have indefinite growth. That is, this is why two cells are used for production of hybrid cells. So, what is a monoclonal antibody? As I have mentioned in the last slide also, they react with a specific epitope on given antigen. Okay, but the polyclonal antibodies, they react with various epitopes on the given antigen. And what is a hybridoma technology? So, there are some B cells, okay, and this B cell B lymphocytes or the B cells, they are fused with the myeloma cell, and this called the hybridoma cells are produced. Okay, so I was where I was, yeah. So, the fusion of the B cells and the lymphocytes, fusion of the B lymphocytes and myeloma cell, they give rise to what is called the hybridoma cells. Okay. Now, this is cultured in a Hath media and the test each wells with for specific antibody, and the screening of this hybridoma cells, they give rise to a propagation of the positive clones. And this is when these are actually these are cultured, this propagation of the positive clones are cultured in vivo and in vitro, and monoclonal antibodies are formed. So, you do not need to remember it in detail. So, what is it in the complete detail, the mechanism you need not to know, but just remember that these are used to isolate monoclonal antibody from polyclonal ones, and how it is done, the fusion of the B cells and the myeloma cells, the B lymphocyte myeloma cells, they give rise to the hybridoma cells. Okay. Now, problem number 17, it says that there is a typographical error here. Okay. So, which of the following statements is false regarding the antibodies? I will just correct it later. So, number A, B cells are associated with the humoral immunity, T cells are associated with the cellular immunity. Number C, T lymphocytes differentiate in plasma cell, and number D, B lymphocytes get maturation in thymus. So, which of the following statements regarding antibodies is incorrect or false, you have to identify. Try to identify it. Options are B cells are associated with the humoral immunity, option B, T cells are associated with the cellular immunity, option C, T lymphocytes differentiate into plasma cell, and option D, B lymphocytes get maturation in thymus. Okay, so let me check if there are any answer for this question. Number B, number D, number D, 3 number D's and 1 number B. Okay, let me check. So, the correct answer is, it is number, okay, it is number D, sorry, I have just not corrected. So, yeah, number D is the correct one. Let me just correct these things. Yeah, it is your number D. Yeah, so number D is the correct answer, very good. So, let us discuss. So, what is an antibody? A blood protein that is produced in response to and counteracting a specific antigen. Antibodies combine chemically with substance which are body recognized as aliens, such as bacteria, virus, foreign substance in the blood. Antibodies, also known as immunoglobulins, as Y-shaped protein that are produced by an immune system to help stop intruders from harming the body. When an intruder enters the body, the immune system spurges to action. These inhibitors, which are called antigens, can be viruses, bacteria or other chemicals. When an antigen is found in the body, the immune system will create antibodies to mark the antigen for the body to destroy. Like B cells, they attack inhibitors the invaders outside the cells, and the T cells, they attack the infected cells. There is a difference between the B cell and T cell. What are the differences? B lymphocytes origination and maturation takes place in the bone marrow, and T lymphocytes origination in bone marrow and the maturation in the thymus. The B lymphocyte differentiate into plasma cell, T lymphocyte differentiate into plasma cell or memory cell or cytotoxic T cells. The B lymphocyte, they are associated with the humoral immunity, and T lymphocytes are associated with the cellular immunity. And the B lymphocyte produce antibody, and T lymphocyte help the B lymphocyte to produce antibodies. So, these are the difference between the B lymphocytes and T lymphocyte. All of these are the examples for the different kind of antibodies. Okay. So, now I will move toward the next problem. So, problem number 18, it says that which of the following statement about intracellular receptors is true. Number A, they consist of 3 protein subunits. Number B, they contain a ligand binding site near C-terminal end. Number C, they contain a binding region for DNA near N-terminal end. And option D, they are activated by hydrophobic molecules which are synthesized within the cell. So, you have to identify which of the following statements about the intracellular receptors is true. They consist of 3 protein subunits. Number A. Number B, they contain a ligand binding site near C-terminal end. Number C, they contain a binding region for DNA near N-terminal end. And number D, they are activated by hydrophobic molecules which are synthesized within the cell. So, let me check if there are any answers for this question, because we need to discuss it also. So, I think few answers, number D, number B, number D, number B. So, I have equal number of D's and B's. So, let me check. So, the correct answer is number B. They contain a ligand binding site near the C-terminal end. So, what are intracellular receptors? They are lipid soluble or small molecule signal pass directly through the cell membrane to receptor. Receptors are almost always enzyme addition to a chemical signal, either inhibits or activates the enzyme activity by altering the shape of the protein. Affected enzymes may be in the cytoplasm or nucleus DNA transcription. For example, nitric oxide activates the enzyme guanylyl cyclase, which catalyzes the production of cycling guanosine monophosphate, causing relaxation of smooth muscles around the vessels, increasing blood flow. Intracellular, some receptor proteins are intracellular, found in the cytosol or nucleus or targeted cells. Small or hydrophobic chemical messengers can readily cross the membrane and activate the receptors. Examples of hydrophobic messengers are the steroid and thyroid hormones of animals. An activated hormone receptor complex can act as a transcription factor, turning on specific genes. So, this is a particular example of a intracellular receptor. Here is a N-terminal end and here is a C-terminal end. So, in the N-terminal end there is a transcription activating domain followed by a DNA binding domain. Then we have a ligand binding domain near the C-terminal end, and inhibitory protein they can bind in this particularly C-terminal end. Okay. And inactive receptor, this is the example. This is a co-activator protein. This is a ligand which can bind in the ligand binding zone, and this is a DNA, this is a hormone receptor. So, this is a particular enzyme, but just remember this inactive receptor here. Okay, and this is the example of active receptor. So, here we remember the C-terminal end near there is a ligand binding domain. Okay. So, we had 1 or 2 correct answers for it. So, now I will move toward the next problem. So, problem number 19, it says which of the following amino acids would be most prone to a reaction with an irreversible inhibitor. Your options are number A tyrosine, option B threonine, option C histidine and option D lysine. So, which of the following amino acid will be most prone to a reaction with an irreversible inhibitor? Try to answer it in the chat box. So, is there any answer in the chat box? Number A, number C. Okay, let me check which is the correct answer. So, the correct answer is actually number D, lysine. So, let me show you the structure. This is the structure of lysine, this is the structure of tyrosine, this is the structure of threonine, and this is the structure of histidine. Now, what is the irreversible inhibitor? In irreversible inhibition, the inhibitor binds to the enzymes irreversibly through the formation of covalent bond with the enzyme, permanently inactivating the enzyme. Okay, so as you can see from the structure for these amino acids, okay, if you look at carefully at the side chain, this lysine have a NH2 group here. The tyrosine, it has a OH group here, threonine and histidine. All of them have in the side chain, they have a lone pairs. Okay, like here the lone pair is on nitrogen, here on oxygen, here on oxygen and this phenolic group, and here is on the histidine. But here in the case of histidine, the lone pair is involved in the aromaticity of this imidazole group. So, this is not that much of nucleophilic for making the covalent bond with the substrate. Okay. For threonine and the tyrosine, first of all, the hydroxyl group is not as much nucleophilic as the amine groups, and moreover this hydroxyl of the tyrosine is involved in the resonance here. So, it is not again in not nucleophilic that much. Only for the lysine there is a free NH2 group which is not involved in any kind of resonance or any kind of aromaticity. So, it is the strongest nucleophile which can react as a react for formation of the covalent linkage for the irreversible inhibition. So, what is the difference between the reversible enzyme inhibition and irreversible enzyme inhibitions? For the reversible one, the process of binding inhibitors to the enzyme through monovalent interaction, so that once removed they allow the restoring of the enzyme function. The reversible inhibitors bind to the enzyme through the non-covalent interactions such as hydrogen bond, hydrophobic interaction, ionic bonds in reversible enzyme inhibition. The enzyme inhibitor complex dissociated quickly, enzymatic action can be restored. The types are competitive, non-competitive, non-competitive and mixed inhibition. Okay. And for the irreversible enzyme mechanism, the process of binding inhibitors to the enzyme through covalent interaction, so that the dissociation takes long time, permanently removing the enzyme action. Irreversible inhibitors bind to the enzyme through the covalent interaction, which modify amino acid residues by reactive functional groups in the irreversible enzyme inhibition. They dissociate very slowly in the irreversible enzyme inhibition. It takes a long time to restore the enzymatic action, occurs through the covalent inactivation of the active site of the enzyme. Okay. So, these were the examples for the reversible and irreversible enzyme inhibition. Now, last question for today's session, it says which of the following agent does not have any effects of its own, and your options are number A, agonist, number B, antagonist, number C, partial agonist, and number D, inverse agonist. So, which of the following agent does not have any effect of its own? Is it agonist, is it antagonist, is it partial agonist, or is it inverse agonist? So, all of these are a type of receptors. Okay. These are all involved or related to it. So, try to answer this question in the chat box. Which of the following agent does not have any effects of its own? Okay, let me check the chat box. So there are answers like number B, number C. Okay, let me check and then discuss all of this. So the correct answer is number B, antagonists. So there are different kind of drug receptor interactions. There are various terms. Okay, agonist is an agent which activates a receptor to produce an effect similar to that of the physiological signal molecule. Inverse agonist is an agent which activates the receptor to produce an effect in the opposite direction to that of the agonist. Antagonist an agent which prevents the action of the agonist or receptor on a receptor or subsequent response, but does not have any effects of its own. Partial agonist an agent which activates the receptor to produce submaximal effect, but antagonizes the action of the full agonist. And ligand, it is any molecule which attaches selectively to a particular receptors or sites. The term only indicates affinity or ability to bind without the regard to functional change. Agonist and competitive agonist are both ligands of the same receptor. So this is all about the agonist antagonist. So antagonist does not have any effects of its own. It only prevents the action of an agonist on a receptor or subsequent response. Okay. So this was all about of today's session. So today we discussed more about the enzyme inhibition. We discussed many the enzyme kinetics methods like the Lineweaver-Burke plot, Hansfield plot. We discussed mathematical questions also. So I hope you understood what we discussed today. If you do not, what about the cysteine in 19th question? Okay, let me show you the 19th question. Okay, cysteine, if you just remember what is the structure of cysteine, in the cysteine we have the thiol group, right? Cysteine is the thiol containing the particular thiol containing. So in cysteine, the sulfur group also it can actually bind with the irreversibly, because the thiol sulfur, it is its nucleophilicity is much better, I think, but if you compare with the other amino acids, cysteine is not that much of the nucleophilic, the thiol group. Okay, so it may be involved in the particular irreversible inhibition. Okay, since it was not in the option, that's why I didn't discuss it. But you can if there is a cysteine in the option and lysine is not in the option, then you can just write it as a cysteine, because lysine is the most prone. Okay, lysine is the most prone, and then we can say the cysteine, there is some probability of the irreversible inhibition, depends what kind of the substrate its work on. We all know that cysteine or the thiol, it actually involves in the disulfide bond formation, right. So if in the particular enzyme there is a thiol moiety or there is another thiol moiety, and if then the cysteine is present in the inhibitor, there will be a disulfide bond formation, which is another method for the irreversible inhibition. Okay, so that depends on the enzyme to enzyme, it varies. So just like every day, I will just share my email ID with you. Okay. So this is my email ID which I have shared in the chat box. If you have any doubt or any query regarding today's class, you just can reach me through the email. I hope you like today's class, and what we discussed, it will help you in understanding the course better, and also for the particular assignment solving. Okay. So today it was the week 3 live session where we discussed the week 4. Next Tuesday from 6 p.m. to 8 p.m. we will have our week 4 live session where we will discuss your week 5 course content. So for now I will just take a leave from the class, and I hope you liked the class today. Thank you for joining, and again happy Independence Day to all. I hope you will attend the next classes also. Thank you for joining today. So I am closing the session for today. If you have any doubt, any query, please feel free to ask me in the email. Okay. Thank you all.